Hi, I'm Kieren.
And I'm Riku.
And welcome to Artificially Ever After. For today's episode,
How AI understands your DNA.
Okay, hi everyone.
Hello, welcome back.
Now, I know I always say this, that we've got a very special episode today.
You do always say that.
But the boy who cried wolf is definitely coming to mind here. But we really do have a special episode today, don't we?
We actually do.
Yeah, so we have interviewed someone for this episode who we're going to introduce in a second. But they're going to be helping us to answer this question and explain how AI is helping within science to read and understand your DNA and understand things about your genes. So this is part of our mini-series that we're doing. We're in collaboration with AIBIO-UK. And our first episode in the series, How AI Unlocks the Secrets of Life, I think gives quite a nice context to the use of AI in science and biology more broadly. So if you haven't checked that out, it can give some really nice context for this. But yeah, what are we talking about today, Riku?
Yeah, as we've mentioned on this podcast, that all AI methods rely on the data set that they're trained on. And as we mentioned in the first episode on how AI is unlocking the secrets of life, we're seeing these amazing applications of AI models using biological data.
Yeah.
And today we're doing a deep dive into one of the most complex biological data sets ever created. And just to give a bit of an insight on this data set, it's about the human genome. So looking at the book that describes everything about you.
Yeah, the code of life.
Yeah, the code of life.
And yeah, we have the perfect guests to talk about that and give us their personal insights. So today we're going to be interviewing Professor Ewan Birney, who's had an incredible scientific research career, has contributed so much to this area of science and just to scientific research more broadly. So currently, they are the executive director of the European Molecular Biology Laboratory, so EMBL, and non-executive director of Genomics England. So they know a lot about genes. So those are their current positions. However, what he's actually known for mainly is his role in the Human Genome Project, so sequencing the human genome and DNA for the first time, which has been described as, the most significant scientific endeavor in human history undertaken in the early 2000s. And his role was to use computer science and design algorithms and bits of software which help to understand and uncover the insights within this DNA sequence. So within this enormous sea of letters, which we're going to talk more about this shortly, but within all of this information that came out of the sequence, his job was to find the bits which encode for genes and which have all the useful information for making us who we are. So yeah, that is our guest today, who we're going to be picking the brains of. Do you want to give a quick overview of what we're going to cover in the episode, though, and then we'll get cracking?
Yep. In the first half, we'll just cover a bit more about DNA to give a bit of a primer, some background knowledge, and specifically a bit of the context around the Human Genome Project. And then we'll talk about the big impact of more compute, more data in this biological space, which drives our second half, which is focusing on the AI machine learning application of this genomic data. And as always, we'll touch on the ethical considerations and a bit more about the road ahead.
Yeah, which are big in this topic, right? I think there's a lot of stigma around sort of genomics and all the stuff you can do with the data, but we're going to demystify it all today and get into those topics, aren't we?
Let's do it.
Okay, so the first big question, in order for us to talk about any of this, what actually is DNA?
Over to you and Bernie.
Yeah, I mean, there's the first thing is just to say what DNA is, and it's a chemical. So it's a very long polymer. So a bit like a hydrocarbon or plastic or something like that. You could write it out as a really, really, really long chemical. Now that would be exhausting and not very easy to handle. It's not how we think about it. And that's because the polymer is made with only 4 different types of components. So in the polymer, you just have a choice of 4, and they have long chemical names, but we abbreviate them to the letters A, T, G, and C. And so a very good analogy is it's a very, very large book. And in fact, in the Science Museum in London, you can see they printed it all out. It's rather brilliant. So it's like a massive bookshelf, and the font is quite small. A bookshelf that kind of goes up, I mean, certainly taller than this room. That is the amount of information in each of our cells. And probably most importantly, that is the amount of information at our very first cell, the fertilized egg, so-called zygote, which makes us. And so to push the book analogy, the genome is a bit like this incredibly long, huge book. We know it has the information that makes a human. Of course, humans have to grow up. We have to live in the real world. We have to interact with other people. So there's more than DNA in that. But it's a bit like a book in a foreign language. So sometimes I make an analogy with Tolstoy. It's a bit like War and Peace, incredibly long, lots of good literature. A surprising amount of Russian farming. We really need to know about it. So the genome has that same kind of feel. There's a lot of stuff where you're like, really? What is this bit? But there's obviously, it obviously makes us. So it's quite exciting.
Incredible. Wow. So I guess given that most of it's about farming and that we have this kind of small percentage that's functional and kind of translating into things that perform useful functions. I was reading that during your time in the Human Genome Project, you organised a little sweepstake before we knew how many genes and kind of protein coding regions there were. Do you remember what your guess was and what was the closest guess? If it wasn't yours, maybe. I don't know if yours was the closest. But
it was 42,851. Mine definitely wasn't closest. Okay. And there's a good story. It's been written up where, collectively, the community in the 90s felt that there should be, quite a few protein coding genes in the human genome, and the numbers varied between, the very lowest numbers were about 40,000 and the very highest numbers were 100,000. One reason why, for example, was a very small nematode worm, the roundworm, C. elegans, that looked like it was going to come in with 20,000 genes. And so people were like, well, we've probably got more than that. And
you'd like to think.
You'd like to think, yeah. It ended up being that that's not the case. It's very clear that we have about 20,000 protein coding genes. That the number becomes one of these science things where it's about the definition of a gene in how you count it. So that's why I'm using the word about. So the difference is less about do we know where these things are, and more about, well, are we going to call this a protein coding gene, or is this 1 or 2,
right? I see.
Or 6 or whatever, and there's some kind of weird scenarios as always in biology. So yeah, I did organize this sweepstake, and basically everybody over predicted, and we actually awarded it to the person who gave the lowest estimate. They were the closest, but one of the reasons why they're closest, or we actually split it 3 ways, because the third lowest was this French colleague, Hugues Rost-Crelius. And he was the person who really sort of advocated for a low number from first principles, from good analysis. And I remember thinking, Hugues, this is very brave.
Yeah.
Are you sure you're right? That doesn't make sense. So yeah, that was a fun thing to do. And just a bit of reflection there, one of the things it taught me, is very clever people can be way off, can be wrong. But the other thing is there was a very big difference actually about trying to estimate something where we didn't have data versus estimating something where we did have data. And once you had data, you could start making good estimates. And kind of Hugues was the first person to make a really good estimate.
Wow, yeah, so I'm conscious you did just mention that the definition of a gene can vary, but I found some research saying that about 1 to 1.5% of our genome are genes, and wondering if you could demystify the other 98%.
Yes, so, and just to be kind of pedantic, it's an important piece of pedantry though, it's protein coding genes, that 1.5% is protein regions that form proteins. And so we've always known that that was always been known that there's a large amount of genome that doesn't encode protein. And one thing is that there's a kind of effectively a genome parasite. Nearly all genomes carry these genome parasites, and they're given slightly different names. So transposons, retrotransposons, and other things. And those parasites are just copying themselves in the genome, and our genome has quite sophisticated machinery to try and suppress the copying. But there's a kind of constant war between the parasites that just want to make more of themselves and our own genome, a kind of genomic immune system that is trying to push it down. But even then, it was a bit unclear what was going on. So I was part of a project to really effectively throw the kitchen sink at the rest of the genome. And there was a bunch of technologies, of which undoubtedly the most successful was something called ChIP-seq, to work out what was going on. And I think the surprise for many people is the genome is very active. So there's a lot of things going on in the genome. Different proteins are binding in different places. Bits of RNA are being made a long way away from protein coding genes and not obviously associated with protein coding genes. Some of them are, but many of them aren't. Now, we kind of knew about that, but I think it was a big surprise to see so much activity. And, we've got used to it, basically. Not all of the activity is necessarily, inverted commas, doing something important. But that's, of course, your definition of the word important. So sometimes there'll be something that, gets activated in cancer. And then when this region gets placed by next door to this region in cancer, then this region that previously wasn't doing anything, inverted commas, drives this cancer gene to make it happen. So it is a big, complicated place, the genome. So there's a lot of information, and then on it, there's a lot of systems that are trying to decode and manage and handle the information in each of our cells.
Wow. Yeah, I love that analogy there about the book. It just really gives some intuition. I think we need to go and have a look at that book now. That's really surprising that I've never seen this because I've been to that museum. But we just wanted to quickly add to what Ewan was just saying and allude to the fact that our understanding has actually evolved quite a bit through time, pardon the pun there, from what we used to think about one gene, one trait. So, when we've identified those genes and those protein coding regions within DNA, we thought that would just associate itself with one particular function and therefore one output in one human versus the other. However, that understanding has evolved to this idea of very complex networks of the genes and their interactions within DNA, and the fact that context, both within the biology of the organism and within its external environment, have a significant impact on what that section of DNA is doing, and it can be kind of interpreted in different ways. I read this really nice analogy which helps give some intuition around what we mean by that, is that if we talk about DNA as if it's like a template for life and for all the functions of an organism, we used to kind of think of it as like a mold in a factory for a part of a car, for example, which, we have this particular mold, we pour in molten metal or plastic, and we get this part out. And the mold can do that thousands of times, and unless something kind of goes critically wrong, we get the same identical car part every single time.
Oh, that's really cool. So with this car part analogy, is that the idea where your genes kind of set your fate? Like you can't fight against your genes, like whatever they are determines what you will look like and become?
Yeah, I think that's the fundamental understanding that's changed, in that the regulation of those genes and what they actually produce in terms of functions isn't always the same just because of the code, and there's a lot of context and additional steps beyond that. And I think the classic example is if we think of identical twins who have that exact same blueprint, all that same code, but they can end up with sort of different physiological traits and things like schizophrenia in one twin and not in the other. So this is a good example that's commonly used. But our understanding has evolved now to this really nice analogy of DNA being more like a script. So if we think of Shakespeare's Romeo and Juliet, for an example, there have been multiple depictions of Shakespeare's initial writings in plays and films. And if we just think of the example, the more recent film with Leonardo DiCaprio, is a very different interpretation of that same text, that same language and writing. And this is the kind of intuition that we now have with epigenetics, that we have identical starting parts, but different outcomes.
Right. And with the Human Genome Project, was that an endeavor to understand the starting parts?
Correct.
Right, okay.
So it's because we previously didn't have a complete map of all of the DNA within that human genome. So we only had segments of the book or of the play script, for example, and we wanted to look at the full script, which would then enable us to analyze and interpret what it was doing.
Yeah, and when I was doing a bit of research on the Human Genome Project, I thought it was maybe a little bit confusing to my non-biology brain. So from what I understand is actually they took the genomic data of, I think, like 27, 26 people. And because actually between humans, yes, everyone has a different like genomic sequence, but the variation is very small. So there's a lot of overlap. So what the Human Genome Project was trying to do was flesh out the sequence of like a general human. Is that kind of how you'd explain it, Kieren?
Yeah, I think so. And just to touch on your point about having different segments there were different research groups around the world which were focused on sequencing different portions of the human genome. And the idea was to then kind of open us up and share them around the world so that we could bring them together and create this full reference genome, which is like what you were talking about there, which we could then compare when people have certain genetic mutations to what this kind of baseline is.
And we're going to hear from Ewan himself about exactly how difficult this endeavor actually was, perceived at the time to be. But we found some really cool quotes as well from the US president at the time, Bill Clinton, back in 2000 when they finally finished this. They said, "Today we are learning the language in which God created life."
Wow.
And even in the UK, we had the science minister say, "We now have the possibility of achieving all we ever hoped from medicine." So
that one was a big claim.
That is a big claim. So yeah, just hope that this gives a bit of this just lays a bit of the energy surrounding this project. So yeah. Over to you, Ewan.
People always knew it was going to be a challenge. So, the Human Genome Project or Genome Project, so going back, 1977, Fred Sanger in Cambridge, UK, works out a new way to sequence to determine the structure of DNA. And it's very efficient. It's very clear that you could do it. It would work. You could only read 500 letters at a time. The human genome is 3 billion letters. Now that's a big difference in those two things. But in the 80s, people realized that it was going to be feasible to sequence the human genome. It was going to take a lot of effort and probably new technologies, which definitely happened. New technologies would have to be developed. And in the 80s, people were doing genetics. Now, for genetics means, there's something, let's take sickle cell, there's a disease and it goes in families, and there is a single change. In this case, there's often just one change in a family, and that single change, when both parents have that same single change, it causes sickle cell disease. And so you can, they were using a variety of, without knowing the whole genome, they could use this tracing in families and some other information about the DNA inside of each families to work out where those single genes were. And so it was clear that it was really going to improve that end of it, and it was going to improve many, many other things around our understanding of the genome, including this kind of, this very kind of obvious statement. If you look down a microscope at a fertilized human egg, a fertilized chimpanzee egg, a fertilized gorilla egg, a fertilized marmoset egg, they're pretty much identical. It's not so different from a fertilized mouse egg. But one thing which is, clearly we end up being different species. We have a different developmental trajectory. There's something in the genome, the DNA, which encodes the majority of the structures that make an adult human. So that was very exciting as well. So there's a lot of, there's many good reasons basically why the Human Genome Project started, both a sort of disease driving one and a fundamental science one. And I was very young then. I was in my mid-20s, and it was known that a big problem was going to be decoding it in all sorts of different ways. And so that's where I was writing, software, you'd now call it machine learning, software to decode the genome, using the bits of biology we knew, and then also kind of effectively doing training style approaches on that. We didn't call it machine learning back then, we just called it bioinformatics, and we still call things which are to do with DNA sequence bioinformatics and protein sequence bioinformatics. But it kind of merges into this modern world of machine learning.
And yeah, I think something to give some context around this time in science and research and history, which is maybe somewhat analogous to what we're seeing today in AI research, is that there was a bit of a race going on between research groups to be the first one to sequence the entire human genome. And there were even some private companies in the US who were saying that they were going to see, if they achieved the sequence first, they would then charge for access to that data. And there was something around this time, and motivated by these different discussions around whether they'd open source the genome and release it or not, which resulted in what were known as the Bermuda Principles, which came out of a conference in Bermuda. And they basically set out to say that any human genome sequence data that was produced and that was captured experimentally should be released and in the public domain within 24 hours of having that data in a research group, which is pretty incredible.
Speedy,
and a fast turnaround as well. So, that was the kind of paradigm shift, and that set the example, because I think that was very much against what was going on in the scientific community at the time, whereas you wouldn't release lots of preliminary data before you'd interpreted it and understood what it meant, and this was a sort of shift towards something much more open.
And just to highlight the significance of these Bermuda Principles, I think Ewan himself said in a different interview that a big result of this was the Human Genome Project actually accelerated 10 years ahead in its original timeline, which had even more consequences, right?
Yeah, well, I mean, Ewan was a massive proponent for open sourcing all that data and making sure it wasn't closed source, as were a lot of others in the research community. But in doing so, in releasing all that data and helping others to kind of sequence and annotate their genome and compare it to the reference genome, massively reduced the cost of doing so. So yeah, just to look at some numbers here, the Human Genome Project itself, in its entirety over many years, is estimated to have cost in total just under $3 billion to sequence one single human genome. And thanks to technological advancements and advancements in techniques in the lab and the data analysis, by 2010 that cost had come down to only a few $1,000s per genome. By 2014 it was down to about $1,000 per genome. And if we look at the cost in 2022, it's come down to roughly $200 per genome, which, not that we can necessarily comprehend it, but that's a 5 million fold reduction from the original cost. And that has now facilitated these really high throughput sequencer companies and factories which can do around 20,000 genomes per year because of the cost and the speed at which we can now do things. So it's been an absolute revolution within the field, and it has facilitated this explosion of data, which compared to what we had as just one genome, we now have in the tens of thousands. We have all these movements to get in the hundreds of thousands of genomes and maybe towards the millions of genomes at some point in the near future. And this is facilitating so many questions being answered. Now we've got this scale of data. And naturally, as we're going to talk about in a second, leads us to wanting and using machine learning and AI.
So now we'll move on to big data in genomics, and just moving straight from what Kieren mentioned earlier about this massive reduction in costs, this has resulted in these huge projects that were previously impossible, but because of all these massive innovations of these AI methods and data collection, we're actually seeing the UK being the world leader in holding these huge genomic data sets.
Nice.
And
big up the UK.
yeah, so it's lovely to see. So Ewan played a huge part in that, and so from 2013 to 2018, so within just 5 years, they actually sequenced 100,000 human genomes.
Wow.
So that was funded by UK government.
Aptly named the 100,000 Genomes Project.
Yeah, they weren't very creative there, were they? And since then, we've got the UK Biobank, which holds over 500,000 whole human genomes.
Wow.
So yeah, and these are also linked with health data. So this is the perfect grounds for AI methods, which are great at finding those crazy relationships between people and their health. So.
And yeah, exactly. This is being enabled because of just this enormous scale that we're now at in terms of the number of genomes we have, and therefore the amount of actual DNA sequence and the number of these building blocks, these letters. Just to give some context on the total number, so looking globally, there was a research paper a couple of years ago that predicted, as of 2025, which we're now in, so it'd be interesting to see how these numbers and predictions stack up, but just comparing genomics and the amount of data they produce to other kind of industries. So if we're looking at big tech, for example, so YouTube produces 500 to 900 million hours per year of video. And that was a prediction for this year. Twitter, or now X, that shows the age of this paper, was between 0.5 and 15 billion tweets per year. So, we're on the order of millions and billions here. But if we look at genomics, this is measured in the number of bases. So each of the 4 letters, building blocks of DNA, we measure it in the number of those, which we are sequencing, which was predicted for this year to be in the order of 1 zeta bases per year. And that is 1 sextillion bases per year, which, can't really wrap your heads around here. But so enormous amounts of data and kind of completely eclipsing these big tech companies that are generating lots of data and people are uploading videos to YouTube. But however, there is an industry which even eclipses genomics, surprisingly, really? Which is astronomy. And in astronomy, the prediction is there's 25 zettabytes of data per year produced. So enormous amount.
Big, isn't
Space is big, so it would make sense, yeah.
But just to maybe put this into more relatable numbers, 1 human genome is about 100 gigabytes of data.
Yeah, nice.
So if you think about your mobile phone, mine has, I think, 128. I could just about fit a genome on there.
Yeah, so interesting. And the crazy thing about that, when you think about it, is that think about the size of DNA and the scale we're looking at with these tiny, tiny molecules compared to the size of the storage device, the SSD on your phone. It's incomparable. So efficiently and densely compact the information in there. And funnily enough, actually, we didn't speak to him about this, but Ewan and a colleague, they were the pioneers of actually storing information and data in DNA, so using it as a storage device. And I think there's a statistic that all of the data that humanity and humans have ever produced, if you stored it in DNA, you could fit in a single room. So it's a very, very compact and efficient way of storing things and information.
Wow. So these strings of DNA are absolutely huge, but are they just useful on their own, or do you need to do a bit more with them?
Exactly. So now this comes to the question of annotation and labeling and understanding all this data, which is where Ewan has played a pivotal role, and all the algorithms he's developed have helped in this area of research and pioneered it, really. So once we have all this data sequenced and the building blocks and the code of different organisms on our computer, we then need to do something with it. So annotation is basically identifying what each part of that genome is doing, and where the genes are, or the protein coding regions. So which bits of that DNA are then read by the biological machinery and then translated into protein, which is basically the matter and the molecules within living organisms which account for kind of all of life, really. And when we're coming on in our next episode to talk about AlphaFold, that's talking about understanding more at this protein level. But yeah, annotation is all about understanding where the genes are within this sea of data, 1 to 2%, as you mentioned earlier, which bits then regulate those genes and control how much they're expressed by and what they're doing, and also which areas or which variations might cause disease. This is a huge question that we'll kind of come on to in the second part, but all of this is under the kind of realm of annotation, and it's part science, part software, so completely enabled by computers and computer science. No human could go through all those 3 billion letters and annotate them manually, so we need computers here and software to automate that process, but it's grounded in the science of understanding what those different nucleotides are and the roles that they play within different organisms.
Okay, so it sounds like annotation's a bit like taking that crazy book that's written in, I'm just going to say Russian, but some language that you don't know, and that no one on earth speaks that language.
Yeah.
And is it the kind of game of looking at the symbols and figuring out what they do? So it's more like, now that we've done the Human Genome Project, we've seen the whole book, but now we need to make sense of it. Is that kind of what annotation
understand what the words are, the sentences, and then learn the grammar.
Right.
And I think Ewan's made this analogy before, and he mentioned that you've got this book, and let's say it was in Russian, but you don't have a Russian teacher. Like we had to teach ourselves the language without anyone understanding the language. So an incredibly challenging task.
So that makes sense then why after you do the annotation, these AI models can really come into play.
Yeah.
Not saying they can't be used without annotated data, but we've seen time and time again that when you give AI models huge data sets and with human supervision, telling the AI what the patterns are, we see amazing results. So
And now back to you, and as someone who I think is perfectly equipped to answer this question as to what was the biggest contributor to these new insights. Was it machine learning? Was it just the sheer amount and volume of data that we now had? Or was it the increase in computational power that we needed to actually analyze the data? Because it's all well and good having the algorithms, but if you're still sat on a 50-year-old computer or really old MacBook or something that can't actually process that amount and volume of data, it's neither use nor ornament. So over to Ewan.
Yeah, I mean, it's really all of the above, and each one of those is a little story. So the drop in sequencing costs allowed us to sequence obviously lots of other humans, and we've done that. And we now have sequenced a large number of humans, so we can see the variation between humans. And you can use that in a variety of ways, both to understand disease, but also understand basic biology. And then we've done it to sequence a lot of species. We now have ambitions to sequence, for example, there is an ambition to sequence all the species on Earth, which is quite an awesome statement. Like a very practical subset of that are all the eukaryotes, so that's kind of bigger organisms, but certainly all the animals and plants and fungi in the British and Irish Isles. So that is that's a slightly smaller set, and that's about 70,000 species. So that's an ambition. There's some way on that. I think they're at least 10% in. So that's not crazy talk. So DNA sequencing, the drop in cost also improved the way we could do analyzed experiments or the sort of scale and scope of those experiments. But then, you get a lot of data coming out. I mean, it's big. It's not as smaller than the Large Hadron Collider, but it's big enough. You can't put it on laptops. You need to have serious computing about it. And there are some, machine learning, not everything will be a simple linear model. You can't, although in fact, when simple linear models work, they work very well. So there's an awful lot of very simple linear models knocking around. But you also want to, understand this. You want to be data driven. You don't want to kind of have to necessarily know the answer beforehand. And that goes to sort of generative models rather than kind of hypothesis testing kind of models, if you see what I mean. So we've definitely done that. We've definitely thrown lots of machine learning at it, and then these deep learning models of different sorts as well. So be it convolutional networks or attention-based networks. So both of those are like a really good fit. And lots of people have been having a great time using all of the above.
Okay, and now time for a quick fun fact break.
Who's going first?
I always try to remember who went first last time, and I don't know. I think you might have gone first.
All right, I'll go first. So my fun fact is about the world's biggest AI chip manufacturer, NVIDIA.
NVIDIA, yeah.
Sorry, I should let you say it, shouldn't I? Right. The origin of their name comes from the Latin word invidia, so not NVIDIA, invidia, meaning envy. And you'll notice that their branding is all green as well, which is the colour of envy as well.
Oh, wow, that's nice. Yeah, I had no idea about that.
Yeah.
Not sure what I thought it meant, but...
Yeah, I used to think that the vid in NVIDIA was about videos, because GPUs used to be good for like video editing before the world of AI, but that's actually not true.
Oh, there you go. There's why all the green. I actually don't really like the green colour that much. It's a bit tacky. Anyway, interesting. My fun fact is about an article that was published just this month, actually, in a US medical journal, the Annals of Internal Medicine.
Annals?
Yeah, like annual.
Right.
And it reported this case in which a 60-year-old man had developed bromism, or bromine toxicity, so to the chemical, which is quite a rare thing to see as a clinician, I believe. And the patient basically told the doctors that after reading about the negative effects of sodium chloride, so table salt, he wanted to eliminate it from his diet and eat less salt. And he basically consulted ChatGPT on this, about eliminating chloride from his diet. And he started taking sodium bromide instead, which you should not consume. over a 3-month period he'd been taking it for. And yeah, this was despite reading in the ChatGPT output that it did say that chloride can be swapped with bromide, though likely for other purposes such as cleaning. And yeah, sodium bromide was actually used in the early 20th century as a sedative, so maybe just got really sleepy. But yeah, crazy story. And I think ironically, the output of all of this is that we should be taking ChatGPT's outputs with a pinch of salt and not without. So sorry, I had to throw that point in there. But yeah, I thought that was crazy.
That is really cool.
Nice. Now back to the episode. Okay, welcome back. So, what are we going to be talking about in this segment, Riku?
Yes, we're going to talk about what AI and machine learning is actually doing in genomics
and why we need it.
Well, yeah, exactly. Yeah, what's it doing? Why we need it, and especially how it builds off what we spoke about earlier about this massive boom in genomic data with annotation. So, what we saw is that the algorithms using this genomic data also changed as the data got bigger, and it's kind of the classic from the more traditional methods, which I think Ewan mentions is like the random forest or the SVM, a linear model. These are more simple mathematical algorithms.
Which are sort of just tools, aren't they, for within this machine learning umbrella category?
Yes, exactly. And I think we spoke about it on our previous episodes where we differentiate between these traditional machine learning methods and deep learning. So this is stuff like the deep neural networks where they're actually trying to model how human brains work. And I think Ewan mentions the attention net and the CNN, the convolutional neural network.
And these are the approaches that are much more data-driven, where what we actually build is just the framework for which learning can occur. So that's why we're trying to mimic a human brain, for example, with a neural network. And then the data is fed through that to actually learn the interesting things.
Yeah, exactly. I think a nice way of summarizing exactly what traditional methods are and these more advanced ones are is that with the newer methods, it's like growing a plant, and you set this algorithm, you give it loads of data, and it grows into this clever neural network. Whereas with the traditional methods, you had to spend a lot of time thinking which algorithm, what are the settings, and you had to do a lot of work before you press train.
Yeah, so you build the plant rather than building the kind of soil and the infrastructure to grow the plant.
Exactly, yeah. So we asked Ewan about whether there was a crucial point where this data got bigger and bigger and these traditional methods just were no longer the solution. So let's see what he had to say.
I mean, I think there are still a bunch of problems that get solved by simple statistics. So I really don't throw them out. But definitely there have been a bunch of scenarios where you're like, well, that doesn't work. What do we do now? Yeah. And very often in biology, you can be in a situation where you say, well, definitely the system works. The egg becomes a human being, the cells make these decisions, whatever the criteria is. And these are the measurements, these are the molecules that we think are involved in that. So surely, how are we going to make sense of that? And for sure, these now, whether we call this deep learning or AI, I don't really care. Let's call it AI for the moment. But these very large, neural networks, that has been tremendously impressive. And in biology, we do a lot of image analysis, and convolutional networks have just been really, really useful. So of all different sorts and shapes and sizes, and I wouldn't be able to list them all. But then these attention-based systems have been, really, really good as well. And you can use them on the DNA sequence, but you can also use them, to do some of these other bits of decoding for this. I have my favorites at the moment, but the most important thing to realize is that there's a big, big kind of space for this. The other thing I will recognize, which I think is really interesting, in AI, it feels a lot more experimental to me. And in fact, there isn't a good theory about what is the right architecture to choose. And there isn't a good theory about quality of the data or stuff like that. So there's an awful lot of like throwing stuff at the wall and seeing what sticks. And that's actually very experimental. And some of the mindset you get from being an experimentalist or working with experimentalists is actually incredibly useful in AI. Like, what's my positive control? How do I know that this thing is even working at all before I launch it on a problem where I don't know whether it's going to work or not.
Let's say we're using these models and AI, machine learning, to learn some meaningful insights that we can then validate experimentally, and we know those predictions are learning something useful in the physical world. Can you say anything about whether they're learning anything that's actually aligned with what biology is doing, or is it impossible to say?
So I think once something which I've grown used to. 5 years ago, I felt these things were very black boxy, and I didn't like that. And I've actually learned now to get more comfortable with what is apparently a black box, but you can look at it and pull it apart. So I think it's actually, it's quite exciting. I think the surprise from people of my generation who kind of grew up with Bayesian statistics and multivariate statistics, and then maybe some traditional machine learning, random forests and stuff, is that you really focus first on making a system that works and trains and generalizes well. You convince yourself that works, and then you study it. Whereas previously you would do a lot of kind of upfront modeling work and then kind of write down the model and try and fit, parameters inside of that model. Yeah. And so there is something very sort of different about the way you order the process here. But it's great. And we should definitely talk. I mean, AlphaFold was my big moment where I was like, I literally fell off my chair and said some rude words when I first heard it from Demis. Yeah. And I was like, oh my God. Because that was a serious problem that had been around since the 1970s. A lot of very clever people had thrown a lot of data and a lot of computers at it, every decade. Yeah. And the step change improvement they made was just awesome. And I still, I think I truly don't understand the network. Lots of things are counterintuitive in that model, basically. But it clearly works.
Wow, that was fascinating and really nice to know we don't just have to throw out all the old methods and start from scratch again. And there is a place for those methods in the data that we even generate today, and we don't just need to apply this new shiny tool to absolutely everything, which maybe seems to be happening with large language models at the moment. But anyway, we're now just going to quickly talk about the types of things and breakthroughs that have happened from applying these kind of novel machine learning algorithms with this kind of much larger data set and better quality data. And the first of which is just starting at that bottom level. So once we've got this kind of annotation and we have this DNA, we want to understand what the activity of those genes are. So as we mentioned earlier, genes aren't just simple on-off switches. We actually have complex regulation, and the genes are controlled by DNA elements that can be tens of thousands of letters away, kind of up or downstream in the DNA, and they actually influence what goes on in that protein coding region. And this is something that's incredibly complex and hard patterns for humans to see by eye or manually annotate. So, and even for traditional machine learning methods, we'd have to say exactly what those patterns were that we wanted to look for. Whereas now, with deep learning approaches, these neural networks can learn these super complex patterns and kind of long-term dependencies within DNA, and they have incredible performance now for predicting how kind of genetic mutations in some regulatory region can alter the gene output or how the gene expression will change. And this is absolutely critical in so many conditions and diseases which happen in these non-coding regions of DNA.
And why does gene activity matter? Is that directly causing like illnesses, and is that helping to predict people's susceptibility and things?
Well, so we know that the coding regions they result in proteins, and proteins cause all the function and the functionality and the operating of living organisms. So we can understand that, if we have a small change in, we've only got these 4 letters, and if one of them flips, for example, or there's some alteration in a very small pattern, that can result in wildly different behaviors and outcomes, which could be a cancer cell, for instance, and something that is growing in a way that it shouldn't do. So yeah, it's trying to understand this complex pattern when we go from the DNA sequence to itself to the gene activity, and then subsequently other things which go on, and all the way to the highest level can affect how you're feeling and what your symptoms are, for example. And it's this complex tree that spans so many orders of complexity and scales within organisms. And the next example of that is when we go a sort of step higher, when we're trying to understand what those proteins are then doing and how those protein structures form. So going from that DNA sequence, we then go to what those molecules known as proteins that encode for all of life, how do they function, and specifically, how do they look in a 3D space? So it's not just about the sequence anymore, it's about their physical 3D structure and how they kind of slot into different parts of the biology and parts of the cell and these kind of binding sites, all these things that now 3D space is much more important. And the prime example of where AI has massively helped here and uncovered so much insight and information into the function of life is with this AI known as AlphaFold, which we're going to have a whole episode on next time because it's such a big breakthrough and such an incredible thing that happened. But it broke this really complex problem, which we thought was unbreakable for so many years, about how do proteins fold, and given the sequence of amino acids in the protein, what does its 3D structure then look like, and therefore what is its actual function and what properties does it have within an organism, because it's a very tricky task to go from that sequence to the 3D structure. And AlphaFold cracked that. So AI did something that no human could ever do here. And we're going to talk about that in the next episode in its entirety, because it unlocked so many things. And there's so much research going on in that area now, thanks to AI. But yeah, what does this actually mean practically and for the outcomes for humans and clinicians and drug designers? Riku, do you want to sort of touch on, what these kind of specific tasks that we can now do mean for humans and for our day-to-day lives?
So this sounds like through AI, we're now massively able to speed up the diagnoses of diseases. Much more personalized medicine, I guess, is the takeaway. I mean, your gene is very individual.
Yeah, exactly. We can now sequence genomes much, much faster, compare them to a reference genome, see what's different, and then associate that with the different behaviors or symptoms of a certain person with a genetic mutation, for example.
Right. And then with stuff like AlphaFold, it sounds like massively accelerates like discovery of the properties, and you don't actually have to do the experiment in the lab because the AI learns it for you. Is that the idea?
Yeah, and then predicts the structure. You don't have to go and check the structure's correct. We can then design the protein sequence for the particular structure that we want, which we know is going to kind of cure the certain disease that we have. So all of this that we can now do computationally, whereas previously it would take years and years in a lab and lots of experiments to kind of quantify things, check what the structure was the sequence correct, which AI is just speeding up massively.
So it sounds like AI is massively speeding up the diagnostic process. But whilst it is super helpful, it doesn't sound like we should necessarily replace experimental validation because AI outputs aren't always correct.
Yeah.
And there's also all kinds of ethical problems in this. So over to you and to see what he thinks on this.
I am worried. I mean, I'm worried about a number of things around AI. So one is what you mentioned, this kind of hype cycle thing, where I actually think the word AI now has become, for many people, just about chatbots and ChatGPT and generative pre-trained transformers or whatever. And I just, I think people are, I think some disillusionment has already happened. Some, it's come in, and I think there's going to be more. And actually, if we're not careful, people will kind of throw the whole thing out, which is crazy, because clearly some things work so well. We are never giving these things up as good techniques, be it AlphaFold, but also all the image analysis stuff, which is just really transformed image analysis. Yeah. So those would be two good examples, but there's many more, and I think some of the genome decoders as well will go into that bucket. So there's this overhype problem with the consumer-facing AI not really coming to things, and then people will be kind of anti-AI, and I think that will be a bit unfortunate. And then you mentioned this problem with medical stuff. We do have to be very careful now. Actually, we've always had this, and all of these questions, like, have I really tested it in a way that I'm confident generalizes well? How do I know that it won't give me weird results? Which, of course, in ChatGPT world is we call those hallucinations, but in this other world, you really don't want that to happen. So I don't think AI fundamentally changes the concerns, but it definitely makes us have many. It makes it a much bigger part of the problem. Like, we've really You've really got to have good criteria, good systems. And the simple thing at the moment, which many people do, be it self-driving cars through to cardiac analysis, is you put a human in the loop, right? So you've got to put a human in the loop. That causes its own problems. Humans get bored if they're just there saying, yes, 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 you're correct. Yes, 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 you're correct. So there's a whole business of like keeping the human's brain being used in the right way. That how you combine the ability for the human to spot the kind of out of distribution, weird scenario, those sort of problems, which clearly humans will, I think, be in a much better place than many of the AI schemes, with effectively boredom. If the AI is doing all the routine stuff and stuff like that. Now, final one, which has also been around for a long time. Indeed, you can imagine making nasty proteins, new toxins, the stuff. I mean, actually, you don't need AI to find a nasty toxin. I mean, there's plenty of natural nasty toxins that you could do, plenty of very nasty viruses and bacteria. So the kind of but what AI does give is a kind of bigger space to explore, harder perhaps to detect. We don't know. So I am that kind of bioweapon, dual use bioweapon-y sort of stuff. Again, I don't think AI fundamentally changes what is going on, but it kind of definitely changes the kind of shape of the problem. Even the problem was always there. Anthrax and nasty viruses that you could be around. But now there's a kind of different shape to the problem, and we have to take that seriously. We can't just say, okay, let's ignore that for this. Yeah.
Maybe that answers my next question then, but I'm really curious because I know throughout your career you've been a massive advocate for open sourcing, and obviously we had the Bermuda principles and that DNA sequence data that you're generating. You thought that should just go straight out into the public domain. Do you see a modern version of the Bermuda principles, or has anything changed now in this era of AI?
Yeah, so I'm very proud of all of that open stuff. And when I think science has always worked best shared and in the open, globally shared and things like that. There are some differences with AI, and AlphaFold 2 was really made fully open, which is great, and we were a big part of that. AlphaFold 3 was made less fully open. It's a bit more complicated. You can't tell commercial companies what to do. That's not the way our society is structured. So it's more important, I think, for the public domain to know what we want to achieve. And we're kind of the tide that raises all the boats, and then the boats sail in different directions and stuff like that. So again, there's been this very long-standing question about what do you do closed and what do you do open. I mean, this predates software. It's kind of arguably a scientific revolution question. The 17th century, and all of that era question. But yeah, it's got this modern twist with this. What are the weights? When you open up an AI model, what do you really need to open up? Do you just open up the pseudocode? Do you have the GitHub? Do you supply the weights? What does open mean in the context of an AI model? And again, keep tuned in biology at EMBL-EBI. We're gonna provide some better ways of tracking fully open models and fully open things coming out in the future, so that again, this will kind of be the tide that lifts everybody. And then of course some people will keep things closed, and that they have every right to do so. And myself and people can't tell people what to do about these things. So it's more about allowing people to make choices about what they think is the best way for their innovations to be realized.
Okay, so the final segment, we're going to start talking about some of these crazy questions which now come about, and I think jump into people's minds when they think about genomics and genetics. And I think it's super important to balance these breakthroughs in AI and applying newer techniques to more and more data, better quality data, and covering all these insights. And like you mentioned, though, if we're kind of handing more control over to the AI and that's facilitating more things, we need to start asking these ethical questions. And I just, doing the research for this episode, actually sprung to mind this film, which I watched a number of years ago, which was really, really good. And it's called Gattaca. And it basically it's a 1997 film, and it's a dystopian sci-fi which basically presents this future society that is driven by eugenics, so controlling how reproduction occurs within a population to prioritize the genes and the genetic traits which persist. And yeah, it's basically where children in this world are conceived through genetic selection to ensure that they possess the best hereditary traits coming from their parents.
So is that like only letting the tall or muscular or smart kids reproduce or something like that?
I think no. So I think in the film, if I remember correctly, everyone can reproduce as normal, but certain people that pay or are in a certain class of society, they can use new advanced genomics and genetic technology to only select the best traits from your parents. So let's say one of your parents had some genetic disorder that wasn't preferable, they could kind of get rid of that, and only the desirable traits would be combined in the perfect way.
Design a baby.
So yeah, exactly. So over time, you then converge on that kind of class of society that use this method. They would converge on being the optimal humans. And yeah, in the film, that was exactly what happened. So an individual's social standing and their opportunities, their job prospects, they were all determined by your genetic makeup. So they had instant sequencing, and it could tell exactly what you were made up of, and you were therefore put into certain pockets of society according to your sequence. And basically, it's a great film. I'd suggest watching it. But the protagonist basically was conceived naturally, so didn't have any of this selection process. And they were classified as kind of invalid due to their genetic predisposition for certain jobs. I think he had a heart condition or something. Right. And yeah, he faced discrimination, and he basically had to go and do all the menial work and couldn't do any of the kind of intellectual tasks. And he really wanted to basically be an astronaut and go to space. But he wasn't within the genetic category for people that could do that. So it's a great film.
That's crazy.
But the question is, with these advanced sequencing technologies, it's speeding up. Potentially we can do what they do in the film, where they kind of put their hand down on a scanner, or it takes a tiny bit of their blood and they know their sequence instantly. All these things are becoming possible. We're able to understand what genetic traits lie within genomes. So this is just a movie, but could these things be possible in coming years? Technology might be there, and we now need to ask ourselves, or start asking the questions as to what would we allow and what would we accept.
Because I guess the question of whether it's possible or real is about how much faster can we make things? Because we're already able to do a lot of those things. So that's very scary. And I guess that leads perfectly into our next point, which is about privacy and identification.
Yeah.
So clearly, if we had this ability to perfectly figure out what diseases you're susceptible to in your whole future from your genome, you're likely not going to want to make that very public. And you might be thinking, well, why don't you just take your name off your genetic data in this big database? But studies have shown that you can actually re-identify their surname, and therefore, not just yourself, your whole family.
Just from the DNA sequencing data, yeah.
Exactly, yeah.
Yeah, and I think that actually reminds me of a famous forensic case as well. So the famous Golden State Killer in America. And this case was basically solved by comparing DNA and data from the crime scenes to publicly available ancestry databases where people upload kind of saliva or bits of blood to these companies that then trace your ancestry. And they were basically able to identify a distant relative of the murderer and kind of identify them that way. And luckily they did catch the right person, but this also raises the question, like you were just saying, if it's only surnames or it's only distant relatives, how do we know for certain we're getting the right individual? So yeah, even the anonymized data alone carries really important information about individuals.
And the problem with machine learning isn't just that you can just hide the data set, but if you were to publicly release machine learning models that are trained on this very sensitive genomic data, we've already seen that with these large language models, that they're completely capable of reproducing the data that they were trained on. We need to be really careful about how we release these AI models trained on genomic data, so they don't reveal people's entire fingerprint.
Exactly, yeah. Like the early versions of ChatGPT, you could, if you prompted it in the right way, you could get it to verbatim rewrite bits of like Harry Potter novels or copyrighted data when it should not have been doing that, and that was a breach of the
and
privacy and
imagine
copyright.
someone types your name in and
Exactly, yeah. So these are the type of questions that we need to ask, and we need to think about kind of all the ways that we can protect individual privacy in the data. But yeah, just a quick final one on the ethics, which is a classic that we always come back to, are the biases that are involved in the data themselves, and therefore the biases that the models have. So this is a huge concern as well. And we've seen even in some of these AI models, and because of the data that they're trained on, that some of them are 4.5 times more accurate for predicting traits and gene function for people with European ancestry versus people with African ancestry. So this just shows what the data is comprised of. Again, very analogous to these large language models and chatbots as well, that they're predominantly trained on English language data and predominantly Western views. So we have similar biases within the area of genomics in terms of what genetic makeup is being used to train these models.
Yeah, and Ewan's actually got a really nice blog piece on this, but why this bias is so concerning is that some people are doing embryo selection based on the genetic data that is gathered in the IVF. So if you're going to select a life from the prediction of one of these models that's trained on this biased data, that's clearly got huge ethical consequences.
Yeah. So when people are using and performing this polygenic embryo selection, where they use AI to predict potential mutations in the genomes of the embryos they're selecting, they literally screen them for their risk of these certain polygenic diseases such as breast cancer, hypertension, diabetes, schizophrenia, and make sure that they select an embryo with the lowest risk of those conditions. So this is literally Gattaca. Crazy.
Wow. And I think the core message here is that these AI models are not the ground truth, and
yes,
they don't know everything. And
they're only as good as the data they're trained on.
Yeah, and I've heard Ewan say in a different interview that it's not that genomic data and these models can't be used for predicting susceptibility to certain diseases, but what's really important is having a solid scientific understanding that actually supports those predictions. And so it's really important that we have the right frameworks in place and human oversight to actually make these decisions.
Yeah. Okay, so that was an amazing conversation that we had with Ewan. We're so incredibly lucky to have got to speak to him on this topic, the pioneer of the Human Genome Project, and analysing all that data that has led to all of this that has happened in science ever since.
Yeah.
So it's hard to actually state how monumental the impact was that he and the other researchers on the Human Genome Project had. But we hope you enjoyed the episode. We covered a lot there, but that is all things to do with genomics and how AI and machine learning is changing that area of science and unlocking incredible new potential and answering these questions that we couldn't do before. So yeah, our next episode, we're going to be diving deep on AlphaFold specifically. So this is the next episode in the mini-series where we are going to speak to someone at the heart of that research project as well and discuss all the ways in which AlphaFold is revolutionizing drug design and targeted medicine. So yeah, so make sure to stay tuned for that.
Yeah, another huge thank you to AIBIO-UK for supporting this mini-series, and also do check out our blog piece throughout the series that's being posted on our website and also AIBIO-UK's channels. So
perfect. Well, thank you so much for listening, and we'll
catch you next time.